네, 조혜린 박사님 아직 안 오셨죠? 음, 설마 안 오시는 건 아니겠죠? 네, 제가 연락 드렸으니까 잠시 한 5분 정도 더 기다려 보겠습니다. 좀 혼선이 있었던 것 같습니다.
아, 네. 제가 다른 줌
네네.
링크로 들어갈 것 같아요. 네.
네네. 앞으로 학생들도 지금 잠시, 거기도 한 7명 더 있었던 것 같은데, 조정윤 교수님
네,
좀
다른
계셨던 것 같고,
교수님하고 6명
네네.
정도 더 있어요. 네.
네네. 그래서 앞으로 여기 링크로 학생들은 들어오면 될 것 같고요. 그래서 조성윤 교수님이 곧 오실 것 같은데, 조성윤 교수님 오시면 시작을 할까요?
네.
거기는 지금 시간이 몇 시 정도 됐죠? 8시, 9시 정도
네,
됐을까요?
8시
아,
30분 정도
늦은
돼요.
시간
네.
감사합니다.
아,
안녕하세요. 저
아닙니다.
내신경과학교실의 고대대 내신경과학교실의 유용진이라고 하고요. 그래서 저희 세미나에 발표해 주셔서 감사합니다. 그래서 오늘은 잠깐 제가 인트로덕션을 하고요. OT를 가지고 그리고 나서 소개시켜 드리고 발표를 시작하면 될 것 같습니다. 혹시 졸업을 하셨어요? 저희가
아,
조소영
아니요.
교수님, 네, 그죠? 아직 그 PhD 트레이닝 중에 계시는 거죠? 아, 네.
네, 한 달 뒤에 디펜스 하고 졸업합니다.
아, 축하드립니다.
아,
네. 네, 조소영 교수님도 들어오신 것 같아요. 그러면 시간이
네.
이미 6분이 지났으니까
네.
제가 화면 쉐어를 좀 하고 OT를 잠깐 진행을 하고 박사님, 네, 예비 박사님이시네. 소개를 시켜 드릴게요. 쉐어. 일단 화면 잘 보일까요?
네.
네. So, welcome everyone. So, before starting the class, I would like to briefly introduce some guideline and some basics for students. So, our classes will be held online for the remainder of semester in Zoom, which is where we are now. I think there are some some people who are using a different link. So, I think this link will be the correct and that's the correct location. And then this semester we have invited 14 speakers from the mostly United States, who is conducting research on various topics from genomics, bioinformatics, and neuroscience, and cancer, immunology, and metabolism, bioengineering, and industry as well. And all 14 speakers are either mostly postdoc and few PhD students and or professor at leading institute in the US. So I believe this seminar lecture will provide great or amazing opportunities to get some insights into the latest research topics and the results. And total grade is divided into four parts. So attendance take 10%, the questions take 30%, and the midterm exam is worth around 30%, and the final exam takes 30% as well. And first of all cameras must be turned on during the class to be considered as present. And also student face must be clearly visible on camera. And also we have another policy for attendance. So attendance will be confirmed based on Zoom login time at 10:30, maybe except for today. And the students who, if you join between 10:31 and 10:45 a.m. will be marked as late. And the students, if you join this class like after 10:45 and do not join at all, will be marked as absent. And we have another policy for camera. So camera status will be randomly checked during the class by me or the Dr. Professor Sung-Yun Cho. And if your camera is found to be turned off during the check, that would be the point will be deducted. Zero point 0.5 point will be deducted. And the second, all students are required to submit at least 3 questions throughout the semester. So students can ask during the Q&A session following the speaker's presentation. And I or Professor Sung-Yun Cho will call out the names of those asking questions in order. And after class, please submit supporting documents for your questions through this KULMS system. I think I uploaded some information in this KULMS in announcement function. And finally, here is the information about the midterm and final exams. And there will be a midterm exam at the end of first 7 weeks of seminars, and the final exam will be after all 14 seminar classes. And each speaker will submit 2 questions. So basically, the midterm exam will consist of 14 questions, and the final exam will consist of 14 questions as well. And we will have an in-person final exam and midterm exam at Anam campus. So students are advised to adjust their personal schedule accordingly, such as like conference or lab meetings. And if you have any question, feel free to email us anytime, or you can submit questions through KULMS system as well. I think that's all for introduction. So today, let's move on to the science. So 박사님 스크린 쉐어 해주셔도 될 것 같아요. 제가 간단히
네.
박사님 소개해드리겠습니다.
네.
So, okay, I'm very excited to introduce our speaker for today, Yelin Cho, who is currently a graduate student in the Department of Material Science and Engineering at MIT. And before that, she finished her undergraduate studies at KAIST in 2021. And then she moved to the US to start her PhD program at MIT in the same year. And I realized that she already has been doing some really great work during her PhD training. And I know she just published one Nature Communications paper last year, and there are 2 other papers is waiting to be published from last year in bioRxiv. So today she is going to be talking about how AlphaFold, maybe AlphaFold 3 style structural prediction models can be used for protein design. So please welcome Yerin Cho.
Yeah. Oh, hi everyone. I'm Yelin. I'm fifth year grad student working in Sergey at MIT. Yeah, although my home department is material science and engineering department, I'm currently working in biology department. Oh, by the way, is it a class for undergrad or grad students?
Mostly graduate student.
Oh, graduate students, gotcha. Okay, cool. Yeah, that all like assume you guys are understanding, yeah, the basic things about the proteins and the biology. Yep, cool. So yeah, as the professor already introduced, today I'm gonna mostly talk about the protein design and then like the current design method I developed like yeah across like several years, past years. So yep, let's get started. So yeah, as you already don't know, the central dogma of molecular biology is like composed of like these different like biomolecules like DNA, RNA, and like protein, which is the polypeptide. So yeah, our body is actually mapping all the information about everything in DNA, and then like throughout this transcription and translation, it generates actually the functional protein that can like serves any kind of like functions in our body. So I think yeah, protein itself is very unique because it has a really unique structure, and the function itself is coming out from the structure. Although there are a lot of like disorder proteins, but like most of the proteins are like somewhat flexible, like that can like serve any kind of like functions in our body or like other environments. So the cool thing about the protein is like, the overall structure is really simple because it's all like connected by like just a one by one this sequential like order. So instead of like being very like dispersed or like connected by a different branches like other polymer is like just a single stranded polymer, and then like everything is like just looks like this string of the beads. So this protein is actually like composed of like very different types of amino acid, but like you can just classify it as a like hydrophobic and hydrophilic residues. So the reason that protein is actually folded into the structure as since like some proteins like hydrophilic residues like water, they interact with water, making this polar interaction, hydrogen bond interaction. But the other residues, like hydrophobic residues, are try not to make an interaction with water. So when you put this polymer into water, it actually folded into some unique structure because the hydrophobic region like try to fold into the core part of the protein, but the other hydrophilic parts try to be like exposed to water. So yeah, after you put into the water and the solution, it eventually came up with this unique structure. So as you can see here in this yellow color, the core part is folded inside, inner part of the protein, and then hydrophilic part is like facing the outer part of this protein, like toward the water. So yeah, protein is composed of 20 different amino acids. So like hydrophobic, hydrophilic, polar, unpolar residues, like some amino acid with a very bulky side chains, and like some with a very like small side chains. So these kind of like unique properties can derive different interactions and like different forms of like the secondary structures and everything. Yeah, so as you already learned, throughout this biology class, this protein residue forms this peptide bond, which is like pretty planar, but like throughout this like kind of uh, uh throughout this rotation angles and the pi psi angles like in this peptide bone, you can actually the protein could be like somewhat flexible, so it's not a like just a static structure. It's not a like single solution, but it can like actually fold into the structure like multiple different structures sometimes, and then like it's kind of like flexible. So I think the protein itself is really universal, and designing the new protein is a very cool project because there are a lot of like proteins that are really, really relevant to our biological system. For example, as you can see in this like left top part, this like PD-L1 protein is very important and like immune system. So like PD-1 is expressed in T cell, and the PD-L1 overexpressed in the cancer cells. And then like if it inhibit the immune system, like we need like some way to actually inhibit this interaction between the PD-1 and the PD-L1 to reactivate the immune system. And sometimes like some allergy proteins like also make an allergy effect. So in that case, you want to like neutralize these allergy proteins to remove the reaction. And also a lot of like enzymes are composed of like different like types of proteins interacting with different small molecule in the protein, like peptide. So for example, in this tap protein protease, it cleavage the protein linkage, but this protein can be also re-engineered through this like machine learning model. And then there are a lot of like antibody, nanobody, like VHH proteins engineering problem as well. So as you can see in this figure already, there are a lot of like proteins that can be actually generated from de novo, not from the natural way, like not from um like and not by injecting some antigen to adimers and like get the antibody, or not just like re-engineering the protein, but you can like totally regenerate the new structure and the like sequence using a de novo method. So how is it possible? So yeah, as you're already familiar with, like there are a lot of like models like AlphaFold, like Rosetta Fold, Earth diffusion. These kind of models are like structure prediction model, or also like protein designs model. So the way that like in the past 5 or 6, 10 years, like the reason we can really develop this kind of models is we got a lot of like data sets. Actually, there are like there is a single structure data bank called Protein Data Bank. So every like biologist, structure biologist, experimentalist like filed all of their like resolved structure from X-ray crystallography or like cryo-EM into this protein data bank. So if you look into this structure database, there's almost like 250 K structures. So this structure data set is a like real asset of like this protein engineering, because like as you already know, if you feed the machine learning model with tons of data set with a very diverse scaffolds and the structures and the sequence, the model can learn the feature of this protein and like learn somewhat the physics of this protein, and then like can understand the basic rule of this biological system. So if you look into the structure, there are like multiple of different functional proteins, like membrane proteins, like small or large proteins. So they have a very unique like set of protein data set in this PDB. So yeah, I want to like classify the current existing protein related models into two different pipelines. One is the protein folding model, and the other is the protein design model. So as you already noticed, like the AlphaFold model, like Rosetta Fold model, is able to, given a sequence, it can predict the 3D coordinate of the structure. So you can understand like easily with this scheme and the right part of this figure. So in the right part, if you train the model like with like sequence, but to predict the structure, the model basically learn how to fold this sequence in this like conformational landscape, and how to optimize this energy. So how to optimize this structure compared to the other alternative like structures. So as you can see in the right, like given this energy landscape, the model sees this landscape and try to like optimize the certain target structure, like compared to the other conformations. And the other style of model is protein design model. This is actually, given a structure, like given a 3D coordinate of this backbone structure, you want to like redesign some sequence that can actually fold into this structure. So the model can like, you train the model by like giving this all of this PDB structure, the backbone information, and as an output, you train the model to learn the amino acid sequence. So it basically learned the 3D information about the structure, and at the inference time, it decodes the amino acid one by one at like each position. So yeah, when you refold this design sequence back with AlphaFold 2 or 3 styles model, like you can confirm that, whether this design sequence can fold into the original structure or not. So you can understand this model as like predicting the probability of the sequence given a structure. So as you can see in this left part, unlike this structure prediction model, this protein design model just can see the single conformation, because you're just giving a like single input structure. So instead of like looking at all of this conformation landscape, it just like focus in this specific single conformation and try to optimize the sequence that can get an energy minima. So sometimes this kind of model can have an off-target issue, because this model can see the single conformation. So like sometimes you design a sequence for this structure, but when you fold this structure, it actually folds into this other structure, because like it has a other local minima in the real like conformation landscape. So yeah, still there are a lot of like problems and ongoing problems on this like structure prediction model and the protein design models. So yeah, as I already show, there are a lot of like already solved problems. You can design a very like highly a high affinity binder toward like some like cancer target, like immune target, everything, but they're like definitely a very hard target. You can't like really design a tight binder toward this target, because I think this kind of like targets have like some intrinsically hard interactions and like physics inside of these proteins. So for example, a lot of like the protein design models struggles with designing a binder toward this like multimeric proteins. For example, like a lot of biological like relevant proteins forms the homoligomeric proteins, which means like you have a set of multiple like copies of proteins and actually folds and bind to each other. So as you can see in the left, this TNF alpha, which is like cytokine style like protein, it actually folds into this structure and like make this triangle shaped homoligomeric protein. So to target this protein, you need to really cleverly like design a binder that can bind to the outer part of this protein, but also the protein that does not interrupt the interactions between this protein copies. So it's relatively hard problem like compared to designing a binder toward this monomeric protein. So as you can see in this right, like DeepMind last year published this white paper about like alpha protein method, which is a design tools, and then they had a really high success rate for the other targets but for the TNF alpha, like they got a 0% of the success rate because like this is relatively hard problem. And also a lot of like enzymes also like forms isomeric the homoligomeric interfaces, and like it has a function in this like homoligomeric states. So for example, this isomerase protein, which is like actually involved in this chemical like glycolysis the process, and that the function appears from the interface of these two copies of proteins. So if you want to re-engineer this protein and redesign this protein, you need to care like about not only this single copy of protein, but also this like dynamic of this interaction, everything. So for the enzyme, it's a relatively hard problem because like you need to like care about the stability, binding, and then also the pairwise, like the interaction, the dynamicity of the protein. So for example, like if you sometimes like to stabilize this enzyme too much, it's like too stable enough and then not moving at all. So it has a restricted like flexibility and not functional at all. So yeah, there are a lot of like things you need to care more if you are thinking about the function of the protein. So yeah, I just like briefly gave the intro about why we need this kind of method, why it's not like already solved problem, why we need a better method to solve this kind of problem. And then I wanted to like talk about my recent method that can actually go one step forward to this problem. So yeah, before going into the details of the models, I want to like talk about these AlphaFold 3 styles models. So AlphaFold 3 came out like 2024 November, and then after it came out, like David Baker and then like people from DeepMind got a Nobel Prize because like this is a really great model, and then now people can solve a lot of problems with this model. So AlphaFold 3, given a sequence or you can additionally give a multiple sequence alignment, which is the evolutionary information of this sequence, and then also template of this sequence, like the contact information about the structure itself. Given this information, the first part of the model, the MSA and the pair module process this evolutionary information and the sequence information and try to like get the global information of this protein, how this like residue interacted with the other residue like throughout this like triangle updates information. So throughout this Paraformer module, you can get the global view of this protein structure by getting the contact map. So you can get a like, given the position I residue, position J residue, what is the probability of the I and J to make a contact and to be close enough? And then given this output, you can put it back to the diffusion model, like which is actually denoising the 3D coordinate of the structure. So given this global information about the contact, this diffusion model like started with the random noise, and every step like is try to like denoise this random noise step by step, and at the end of like 200 inference steps, like you can get it like fully denoised 3D coordinates, as you can see in this right part. So with this very clean structure, you can also get an additional like confidence score, because like sometimes the models struggle with predicting the structure. So you need like some method to evaluate whether this prediction is good or not. So you can use this like PLDT score, which is the sidewise score, or PAE score for the pairwise score. So you can see the value and like just evaluate, whether this part is like highly confident from this model prediction or like less confident. So you can also like use this confidence model as a matrix of like your evaluation as well. So yeah, then how can we actually use the structure prediction model for designing purpose? As I already told, you can structure prediction model throughout this training process, it can actually learn this like how to predict this like structure given this like conformation landscape. It can like view all of this like conformation landscape and try to like figure out the most energetically favorable one. So most probable like structure. So it already learns like which structure is good and which structure is bad, which sequence is good or which sequence is bad throughout this like huge amount of training data set. So the model already like internally know which sequence is good or bad. So for example, like if you give a really good sequence to the model, the output structure will look definitely good and the contact map will be very clean and you will get a very high confidence score. But if you give a random like just random like mix amino acid sequence to the model, the predicted structure will be like very bad and the contact map will be also not clean enough and the confidence score will be low. And then for the optimization, you can actually use this kind of like output, the contact loss and the confidence loss as a optimization loss to optimize your input sequence. What does that mean is like in the machine learning, the way you train is you get the loss and then you update the model to actually like minimize the loss, and then like you update the weights to actually get the like the lowest loss at the end. But instead of doing that, you can like update your input. What does that mean is like, for example, throughout this forward pass, you can get a structure, the contact confidence score. You calculate the loss functions throughout all of this information. Given this loss function, you can backpropagate this loss through this gradient optimization method. It's like somewhat mathematical, but yeah, if you understand, for the simpler understanding, yeah, given this like initial X sequence, you get the loss and then you get the gradient, gradient of this loss and the backpropagate through this model and update your sequence. So the sequence is like going toward the direction to minimize the loss. So this is like the whole point. Throughout like calculating those loss and the backpropagating this loss back into the sequence, you're like keep updating your sequence to actually to get a lower loss than the previous step. So if you do this like step for 100 steps, like at the end, you will get like somewhat good structure with a low loss and a high confidence score. So how is it possible? Because like in the machine learning, this gradient optimization is all done in this like smooth logic optimization steps because every value is not a like 1 or 0. Like everything is like the certain value, like soft value and the like smooth value. So you need to gradually optimize this like the losses back into the sequence. But how is it possible? Because like protein is the like it's not possible to get a like multiple residue at the like single position, so it's always like 1 or 0 problem. So how is it possible? We figure out it actually works in the soft logic space, which means it actually works in the like soft Gumbel noise or the like softmax space even more stably like compared to this like one had encoded the single amino acid sequence based encoding. So we actually figure out like, oh, what if like we optimize everything in the soft logic space? So optimize the gradient and the loss really smoothly in the soft logic base, but at the end, like we are forcing the model, forcing the sequence to be a one that encoded. So at the end, like we can get a like single sequence solution. So yeah, it's somewhat like very complicated, but I will like show the scheme that can like make a better understanding. So overall, like pipeline. Before I'm talking about my method, I want to like show this method first, which is Minecraft. I contributed like to this paper first before developing my new model because like Minecraft is developed after AlphaFold 2 came out. So it's a protein binder designing model. So given this target, as I mentioned, you initialize the binder sequence randomly, just randomly. And then like throughout this prediction and loss optimization, you're just optimizing the sequence like step by step, every step. And at the end of like hundreds of different steps, at the end, you will get a highly confident structure that can bind to your target. And you can simultaneously get a structure and sequence together. And then like given this structure, you can fix the interface residue and then like redesign the rest part of the protein, like to make the protein to be soluble. And then yeah, you can like cross validate with other models like to confirm whether your sequence actually folded into this structure. So yeah, this is the whole pipeline of this like kind of like the gradient-based optimization. But the problem with the AlphaFold 3 was oh, actually, AlphaFold 3 is very different with the AlphaFold 2 because AlphaFold 2 uses a deterministic this like the attachment mechanism, but the AlphaFold 3 it introduced the diffusion model. So diffusion model is actually in the inference time you need to decode the structure one by one every steps. So it requires the 200 of decoding steps. But if you do this like gradient optimization based pace, like you need to actually optimize these for 200 steps for every single structure. So it is really, really time consuming. So we were like actually thought about, oh, how can we actually better optimize this sequence instead of like using this fully denoised structure? As I already mentioned, you can gather like global information about the structure throughout this like first stage of this model prediction using this Paraformer trunk. So the output of the former is the contact information. So if you look into the structure, the contact map is like L by L, which is L is the binder length plus target length. So each position is each residue. So if you look into this like black part, which means like, oh, this residue is actually making a contact with this like residue. If you look into this intercontact residue part, this binder residue is making a contact with this target residue. You can figure out that information. So all we need for this like binder optimization is we want to like generate something that can make a highly probable interactions with a highly confident the compact structure of the binder itself. So all you need is to like just optimize this contact map instead of like fully optimize the 3D coordinates. So yeah, just simply saying, all you need is to like just optimize this histogram, which is the predicted pairwise residue distance distribution. So yeah, I want to like just simply show the scheme of this method itself. Like, so given a target, so your target could be everything, like small molecule, DNA, RNA, protein, and you randomize the sequence. And given the Paraformer trunk, you get the histogram, the contact information. So given this small molecule, you get the contact between this small molecule atom and then like this protein sequence, and then oh, just like measure the how confident this interaction itself is, and then backpropagate that loss back into the sequence and then update it until your loss converges. So at the end, you get a like highly confident sequence structure. So yeah, this is the step-by-step optimization. In the right part, you can see how we generate the sequence and structure simultaneously throughout this process. So when you initialize everything, like, yeah, from very soft losses randomly from random Gaussian noise, everything looks like this, like alpha helical structure. And then like you try to like optimize more and more. So throughout this like 4 different stages, like you're forcing everything from very relaxed sequence space to softmax and then like to one-hot encoded states. So you're actually like forcing everything with a very broad solution, multiple different like solutions, but at the end you're forcing it to like one single solution at the end. So you can see that as you go more and more, yeah, it's starting to form like binding pocket around this like ligand and fold it into the compact structure, like compared to first like two helical structure, right? So yeah, this is like kind of magic of the structure prediction model. And then like, yeah, you can definitely, this is like kind of the showcase of showing that this, you can always invert your structure prediction model for designing process. So yeah, I would like talk about some experimental success rate, and then also binders like we experimental validated. So as you can see in this like figure, we purposely designed like some binder that can interrupt this interaction between this PDL1 and the PD1 protein, and we designed like 10 different proteins, and like 8 out of 10 proteins were expressed, and then like 4 out of 10 were showing a binding affinity toward this PDL1 protein. So yeah, most of the proteins, like we purposely designed something that can bind to this like interface. So when we overlap the structure, you can see this like gray part, pink part, which is the binder actually, yeah, bind to the exactly the same interface. of this PDL1 and the PD1. So yeah, this is like pretty cool. And we also wanted to generate like some small molecule binding protein. And then yeah, we purposely designed a 10 binders that can bind to this small molecule. And then yeah, we experimental validated it, but at the end we just got a one single binder that can that shows this fluorescent activity. So this is the fluorescent ligand that can have a fluorescence activity when this ligand is like not flexible and the planar enough, and they'll actually fix into this like planar conformation. So you need to like design a protein that can fix this ligand to be this planar conformation. So when we actually designed this protein first, we didn't know about this protein, the ligand properties. So I thought, oh, if this protein binds to this ligand, yeah, it will have a fluorescent activity. But when I like read the literature at the end, I figure out, oh, it should be like planar enough to be fluorescent. So when we look into our successful design, yeah, it actually has a very planar the structure, and like it actually forms this like pi packing between these like two hydrophobic the ring shaped side chains. So yeah, it can actually maintain these the planar conformation throughout this interaction. So which is pretty cool because so far like yeah, in a lot of like papers, they try to design a fluorescence protein toward this ligand, but it was really hard because it's hard to maintain this 3D the conformation, the planar conformation, and the easiest way to design a fluorophore binder is just design a beta viral protein that so the inner part like can capture this small molecule to maintain this conformation. But we actually designed a like just alpha helical structure that can also serve the same functions. So I think yeah, this is a pretty cool research because like we actually showed that you can design a like totally new the same function with a totally new topology. And we also, yeah, design like some binders for like IL-7R alpha protein and the BHRF1 proteins, which were previously used as a target in the previous like Earth diffusion or alpha proteo paper. So we also like show that, oh, we can generate a like several hundred nanomolar KD binder or like really tight binder, like 2 nanomolar binder, as you can see in the right. Yep. So yeah, in the paper, we also show some in silico success rate. So in silico success rate is basically based on the computation score. So how many design structure pass your in silico like confidence score like PLDT and PAE. And based on that, yeah, we can confirm our model that can generate a very high success rate that can pass this pretty strict filters. And then yeah, I want to like summarize this method as kind of like generalizable binder generation tool is kind of like off the shelf model because we didn't fine tune or retrain a model, but you can always like plug in this kind of like styles, this kind of gradient optimization scheme to any kind of other structure prediction model. Like not only AlphaFold 2, not only AlphaFold 3, but you can also use it for Bolt, Chai, or Pertinax, which are the like recent structure prediction model because they're like basically using a like very same similar structure prediction model, but as I mentioned, all you need is to optimize the confidence loss, like structure loss, and then like optimize your sequence given that loss as an input. So yeah, briefly summarizing my method, given this like all atom, any kind of molecule, small molecule, protein, metal, DNA, RNA, or even covalent modification, yeah, you can totally redesign or generate the binder from the scratch as a de novo way. And then once you get a like structure, you can redesign like the surface part of the sequence using an inverse folding model, which is like given a structure, redesign the sequence to solubilize your protein more. And then yeah, you can always like cross-validate with other model, or like sometimes you can run the MD simulation or docking tools or other like cross-validation tools to just like check, oh, whether it bind to the correct like spot, or whether it makes a like right coordination with a metal atom, or whether it has a like good enough of a like delta G binding interface between this binder one and the target one. So this was so far I explained about the Bolt design pipeline, and now I want to like focus on this like second pipeline. So I thought, our method is really cool and that we can do a lot of like different tasks and decide a lot of different things, but the biggest cons of this method is it's actually really slow because as I mentioned, every single time you need to make a full like structure prediction and to get the loss back propagated. This is the single step one. And then like to optimize the structure, you basically need to iterate this process like 100 times, 200 times to get a one single good solution, which means like even a one GPU, you need to basically run this like process for 10 or 20 minutes to get a one single good binder. So in computational, it's like relatively expensive and yeah, time consuming. So we're like thinking about to like develop the better method. And other comparable like faster method is actually yeah this diffusion based like fine-tune model like Earth diffusion or Boltzmann the recent paper, because like this model is just fine-tuned on the structure prediction model. So they fine-tune the model for the structure generation model. So it doesn't really need to repeat this process at all. Like the within the one single like the forward step, it will like generate everything from the like noise to the fully denoised structure. So given this structure, they always like get the sequence back with the inverse folding model. Given this structure, we design the sequence and get the finer sequence and the structure together. But yeah, the problem of this model is your sequence design ability is kind of like dependent on the structure quality. So this diffusion model like can generate a decent amount of the good structure, but sometimes it generate a very like bad, not good looking structure. So given a bad structure, it's hard to redesign a good sequence on top of it. So you always need to like think the structure and the sequence problem as a joint problem instead of like independent problem. So to solve this problem, they basically just like draw the multiple states. So like for example, like generate like several thousand of different structure and redesign it and then refold with the AlphaFold and the filter out the bad designs and like just choose the rank them based on the confidence score and like get the top 10 or 20. So this is also kind of like time consuming because you basically need to pretty like thousand of different structure at the initial place. So it's kind of like trade-off between this like accuracy versus the optimization time per design. So we're like thinking about, oh, is there any way we can actually improve both the accuracy but also accelerate the computation time for designing with a one binder? Oh, yeah. So just give a brief overview about this, like fine-tuned version of the uh, protein generation model is basically like retrain the model like with this diffusion model scheme. So using a structure prediction model, like with the fully mask sequence, they always like train a model with a full noise. And then like at the end, they are try to like generate the protein structure. So it's kind of like a random unconditional generation given a like a random noise. So what we have developed, before we developed the second method, it's basically like the idea came out from this observation. When we read this AlphaFold 3 paper, there was a one figure about this, and this was pretty shocking to me because um they made a prediction for this like almost 2000 residues protein which is already in PDB, and this protein is a disorder protein. So disorder protein is not a like protein with a very clear like secondary structure and a like 3D all like Glover structure. Instead of like folding into very like clear structure, it's actually just flurrying around. Like sometimes folded into structure, a lot of like time it like just unfolded and like do some function and refold it back into the other conformation. So basically it's really hard to understand the disorder protein because like they have a lot of like flexibility in terms of their structure. So after they developed the AlphaFold 3, they made a prediction on this the nucleopore complex, which is the disorder protein. And they make also compare with the AlphaFold 2 model. So as you can see in the middle, AlphaFold 2 correctly predict this like disorder spaghetti-like structure, right? Given this sequence as an input. But when you look into this AlphaFold 3 structure, it actually folds everything into a very good like alpha helical coil structure. So which is like not true. So we figure out this like model actually kind of like hallucinate. Hallucinate means like making a like fake result, give it a very like um in confidence sequence back into the model. If the model itself is unsure about the sequence, it's try to like fold into like some like confidence structure, because this diffusion model is like usually trained for the generated model. So if training data set is the out of the distribution, it's try to generate like something good looking, but it's actually fake. So to improve this, the quality of the model, what they have done is they purposely fine-tune this model on disorder protein. So they make a collected all the disorder protein from the PDB database, and they made a prediction with the AlphaFold2, and the AlphaFold2 correctly predict this disorderness of the protein and give this structure as a like final product of this model, and then like train the model to actually predict this disorderness of the protein. So yeah, after you fine tune this protein, it started to generate more like spaghetti-like structure for the disorder protein. So basically this paper, the AlphaFold3 paper, oh, we probably solved this problem because like we purposely did that, and then now it doesn't really have a problem. But we figure out the problem still stay there, because this BRCA2 protein, as you already know, like this is a disorder protein, right? So when you make a prediction with the AlphaFold3, it predicts this disorder protein as a spaghetti structure. So which is good because it's this disordered. But when you give any kind of like random binding partner, which is the RAD51, the pink, the protein and the right, it started to form the superiors, the alpha helical structure around these proteins. So even though they're not like interacting with each other in the real world, they started to make this like very nice interfaces, and they're wrapping around each other like this with this very nice interface. So we still we figure out, oh, this model, even though it's fine-tuned on this disorder protein, it still has the same issue when they're predicting this like multiple chain of proteins, and then yeah, the protein with a like target and a binder. So if the interaction itself is like inconfident model, like try to generate something looks good instead of like generate a bad structure. So this is really bad in terms of the structure prediction, but we started to think about, well, this structure looks pretty good. What if we redesign the sequence, like given the structure, that can actually reform it back into the structure. So because like in terms of protein design, it's not bad to get the good structure, because like anyway, you can use an inverse folding model like protein MPNA, ligand MPNA. You can redesign the sequence that can actually fold into your input structure. So what if like we repurpose this hallucination and this misfolding problem just for generating the initial pose of the binder? So for example, given this like protein target as an input and given a random like G repeats of the protein, in the real world, it shouldn't actually generate this like good binder. But when we fold with the AlphaFold3 structure model, it started to fold like into somewhat okay structure, and then like it bind near to this like binding interface and like fold into the like secondary structure. So we try with the other tokens, like instead of like using a real amino acid, what if like we use this like the fake amino acid, which is X? So people train a structure prediction with these 20 amino acid, and additionally they train with this X token, because there are a lot of like unresolved residue in PDB. So structurally unresolved, sequence unresolved, like there are a lot of like not a lot of, but some like known canonical amino acid, amino acids. So the protein that is not like natural 20 amino acid, they replace everything into X. So we thought, oh, X probably also learned the distribution of the amino acid, but it doesn't really have a specific like bias toward a certain amino acid. So what if like we make a prediction with this X repeats of tokens and like structure this target, and then it actually started to generate an even better structure, the binder. Like it can actually bind to this target with a good looking like this helical structure, helical bundle structure. So yeah, like using a Boltz design method, Minecraft method, we needed to actually, yeah, like repeat this hundred of different steps for one single generation. But now, all we need is the 1/4 step of this structure prediction, because like we can use this hallucinate. We can use like random amino acid, like all X tokens. We can like actually generate this some structure that can like fakely bind to this protein, right? So yeah, like after get this structure as an input, we redesign the protein sequence with a protein MPNN. But as I mentioned before, if the structure input is not good, design sequence is also not good, because like yeah, a protein MPNN always train on this PDB data set, which are the good structures. So they don't really know about, oh, like bad structure. So if you give a bad structure, it suddenly, yeah, just design bad sequences. So if I just do this like single step, like structure prediction and redesign this sequence, a lot of times it's just like fall into the bad structure after we refold it with the AlphaFold or three different like cross-validation models, because you are not like jointly optimizing the structure and sequence together. So it is like so far away from the good solution. So what we started we started to think about, how can we improve this design more better? Like, but not changing like something totally from the scratch, but it's how can we actually optimize this interaction and the sequence more and more. So this paper was also like published like last year with the collaboration from our lab and the Chris Frank from Germany, and then they used this kind of like co-optimization from the AlphaFold 2 and the 2 and protein MPNN. So they generate the like initial structure and a sequence, given it to the protein structure prediction model, and then get the per residue level of like confidence score. So if the certain position is not confident enough, they redesign with the protein MPNN. But if certain position is like already confident enough, they just maintain the sequence and they make a new structure prediction and then redesign the sequence. And they repeat this process multiple times, and then at the end they got a like final sequence candidate. So as you can see in this right part, so the gray one is their initial design, and this like pink one is their final design. Although they looks very similar, they have a very like different confidence score. So yeah, AlphaFold doesn't really like this gray structure, but when you repeat this like yeah, structure prediction, redesign multiple time at the end, like although it has a like just a subtle change of the structure by our eyes, it has a really confident structure, and you can actually experimental validate this design fold into this like structure. So which is pretty cool. So in the machine learning model, like although like yeah, by our eyes, yeah, it looks good, yeah, it's okay, yeah, it's good structure, but like the model itself is really sensitive to the amount of the noise. So yeah, this kind of like optimization is really important problem. So I also incorporate the same approach. So once we generate the initial fold with this like hallucination approach, so we repeat this like protein MPN redesign method and like structure prediction again and again. So given this structure as an input redesign sequence, given this redesign sequence as an input, we predict the structure of this complex and then repeat this multiple times. So as you can see in the bottom, as you go more like cycle, as you repeat the cycle more and more, like yeah, it just like try to like optimize the binding pose more and more, and try to like figure out the better binding pose, the rotation, and the better interface, and redesign the better interface as well. So yeah, I want to like just summarize all of this different protein design model into these three different schemes. So the first one is like multi-step gradient update, which is both Minecraft, like our both design model, which is good. Yeah, you can optimize everything really precisely, but the bad thing is it's really slow. The second is like a single diffusion trajectory-based method or diffusion Voltage-Gen method. But for this one, since you're not co-optimizing these two properties, you need to predict the structure with the multiple seeds and filter out a bit bad designs. So yeah, this is you need to yeah, there is definitely the trade-off between the accuracy and the like the computation time. And the third one is yeah, our model, yeah, we can actually accelerate everything really fast, because like we don't need to yeah, like do the gradient optimization anymore. We can like just predict everything in a single force tab, but by repeating the structure prediction, redesigning sequence tab, you can co-optimize the structure and sequence better. So in this diagram, you can actually yeah, get the structure a little better, then sequence a little better. Like yeah, if you repeat this multiple times, it's much more easier to get a like good solution that can satisfy both structure and the sequence probability. So we defined this as a like protein hunter method. Yeah, and this is really fast and refine the structure iteratively really quickly. So yeah, as a computational validation, as you can see in this left, we try to like generate the structure unconditionally, just making a random protein, yeah, with a very different length. So as you can see in this like different bars, as you repeat this cycle more and more and more, you can see this PLDT, the side-wise confidence scores go up. So as you repeat this more and more, you can like make your initial design to be more stable and then more confident. So yeah, as you can see in the right, it doesn't really change the fold at all, but it's like just changing the subtle changes in this local environment and everything interaction. So this kind of like subtle changes makes the structure even better than before the cycling. So as a result, when we look into this interface confidence score from the AlphaFold 3, which is the IPTM score, like given this multiple of different targets, we just like generate the random binders. So we compare with the artificial method, and then in a computational score, we could achieve many designs that has a much higher IPTM score than this Earth diffusion. So as you can see in the right, yeah, we can design a protein binder that can bind to this like beta bitter protein. We can design a cyclic peptide by concatenate the N terminus and C terminus of protein together and cyclize this protein as a peptide. And yeah, we can generate this binder. And also you can like design your the small molecule binder that can bind to your small molecule of interest. Um, and then like the other thing we try computationally is since if you're just unconditionally generating the structure, often the problem is it just started to generate more of like helical structure. The reason behind that is the generation models actually follow the training data set distribution. So training data set here is PDB dataset. If you look into the PDB dataset, maybe 70-80% of the structure are the alpha helical structure. A lot of like natural proteins are alpha helical structures. So if you make this like random like prediction, it's starting to make a lot of like helical structure, as you can see in this left part. So we started to think about, oh, how can we actually debias this helical signal from this generated pipeline and then like generate more beta shift structure. The way we implement it is, yeah, very intuitive way. So if you look into this like helical structure, the most strongest interaction in this helical bundle is position I and I plus 4 in a different helical like location. One is upper and one is downward. So they're like forming this interaction. So the reason they're making this helical structure is position I plus 4 are forming very tight interactions. So in this like histogram, the contact map, what if like we purposely kill the signal in that position? So we purposely killed the signal there, and then we gave the model the negative helical bias in this like the histogram module, and then after that we started to generate the more beta shift structure. So yeah, using this like very simple tweak and like modeling, yeah, you can actually diversify your structures and designs. So yeah, I want to say our model can do end-to-end like protein design, because like given a structure, you can randomize everything and then like design it in each post and refine more and more. But the other thing you can do is like also refining your designs in the lab, like using other models. If you already have your binder, but which has a very low binding affinity or not binding at all, you can actually rescue that design using this pipeline. So for example, like given the structure and the sequence, you can redesign the sequence and then like you reforward that structure and repeat this multiple times using this protein hunter pipeline. And at the end, you can get a like totally new sequence, but more highly probable, and then like generate the sequence that can bind to this target. So yeah, I want to like show some examples how we can actually utilize our pipeline for the failed design from the other method and rescue them to be a good binder. So here for this human riboflavin kinase, this like Bolsjan, the very recent paper, which is like from MIT, and this is really, really popular method, but I think this is like still single step diffusion model, and that you basically need to like run the model to 20, like 30,000 steps to actually generate the few binders. So this is like very time consuming in the academic lab because we don't really have a GPUs. So I just like thought about, oh, what if like we get the failed design from their paper and purposely redesign this design and like actually transform into the binder. So we got this design and repeat this our process, and then like upon this like cycle, you can see there is an improvement in terms of like interface scores and also the sidewise score. So given this sequence, we totally redesigned this like new sequence, but that can actually fold into like pretty much same structure as the input. So if you look into the interface regions, it actually changes the sequences a lot. And then when I look into the structure, overall like structure are pretty same. Yeah, we still have a like similar binder that has a similar interaction, but we actually generate a binder to be we actually transfer this non-binder to the binder. So we now had a KD value 1.7 micromolar concentration and a KD 200 nanomolar for these two redesigned binders. So when I look into these interactions, like previously for the known binder, there was no like specific hydrogen bond interactions between the protein target and the binder, but now we started to form more like specific hydrogen bonds here, like between the target, like although this is like very the flexible regions in the interfaces. Another example is, this human alpha acid glycoprotein, which has a very long, like the C-terminus tail. So this is like also the beta sheet proteins, and like it has a very unique curvature. So also relatively hard to design a binder, but we also use our like protein under pipeline to design some binder that has 1.6 micromolar KD, which is not very tight. I think this target itself is very relatively hard target. So yeah, previous method all failed, but we could design at least like one binder that shows a binding affinity toward the target. And the other thing is like, in the States, we recently had this like Nipah virus competition. I heard that like Nipah virus recently came out again. And then, the way we can innovate this Nipah virus interactions is innovating the interactions between this apirin B2, human apirin B2 protein and this Nipah virus. So you need to like design it like nanobody like or antibody or the novel protein binder exactly to this interface. And the binder should also have a very high affinity to actually compete with this natural binder. So yeah, if the binding affinity is too low, it will not compete with the natural binder because like natural binder has a like higher binding affinity. So it's you need to like consider both a binding affinity, but also, oh, does it actually bind to the right binding site? So those are the two considerations. So after we release everything like as an open source code in our GitHub, like just in the public, like people just utilize our method and design a very cool binders. So yeah, the Sergey and the Max from Denmark and Spain, they utilize our models, redesign a binder for this Nipah virus, and like could achieve the binders that has a 25 nanomolar and then 187 nanomolar KD values. So yeah, this is like pretty cool. And then we can actually generate a binder that can bind to exactly the right binding contact. We didn't do any like neutralization experiment yet, so we don't know like whether this actually neutralize a natural interaction or not, but it's still good. This binder like bind to exactly the right place. Yeah, the other problem, we haven't tested it in the wet lab yet, but at least in computationally, we show, we can probably like solve the multiple interesting design problems. One is, as I mentioned, we can design a multimeric binder protein really quick because like previously, this gradient optimization method, you actually need a lot of compute to predict the single structure. So if you give three different copies of the proteins, is super slow. But now since we are just using a single forward pass, we can just give three copies of proteins and like generate one binder toward it. And the other cool thing is like you can do the multi-motif scaffolding. Motif scaffolding is given this already existing natural binding fold and the like this binding pose, you just extract the active site residue or conserve residues and then redesign the rest part of it. Like that can just support this motif and like improve the connectivity. So as you can see here, like given this heme ligand, you can like just fix the wild type natural like heme binder like around this active site and redesign the rest part of it using a 1300, and then we generate this pink like part that can connect these multiple different motifs together. Yeah, the other is like antibody partial redesign problem. So given this target and antibody, the light chain, heavy chain, you can just like purposely redesign the CDR regions. Like, so given the framework to be fixed, you can redesign the CDR regions to generate the new binder. So yeah, I think yeah, to summarize, the key takeaway is yeah, this is pretty cool, like the all-atom protein generation tools kit, but improving the filtering evaluation is still very important because anyway, yeah, we generate something and always like just filter based on the in silico metrics like PLDT or in silico energy term, everything. And then those energy terms confidence scores are not perfect. So we can't really guarantee whether this filtering will help to increase the experimental success rate. So it's really hard. I think we need to parallelly improve the designing tool, but also the filtering tool to actually increase the well-loved experimental success rate more. And the off-target issues is also like significantly important because in the binary design, you always like optimize for a single target, so it doesn't also guarantee whether this bind to the other target, like by a target 2, 3. And then if you want to like make your binder to be like super specific for this, like your target of interest, you need to remove all of these like unnecessary interactions. So those are also a very challenging part of it. So I think yeah, there are like still a lot of problems we need to think and then we need to resolve and then like yeah, improve this field like with everyone. So yeah, we already preprint this method and then make our code to be available in GitHub and a collab notebook. So I think yeah, anybody like if you have any interest in this kind of projects and if you have like target, yeah, you can like definitely yeah test this our method to design a binder or like yeah new functional like enzymes or binders everything. So yeah, feel free to play around with these tools and then yeah, we're still um making our tools to be better. So stay tuned with that. Yeah, I finally want to acknowledge our collaborators. So this whole design is done with our lab and the other external collaborators from EPFL and then University of Zurich. So Martin Pesea, who is a yeah first author of both Minecraft, and then Protein Hunter is done by yeah like IPD, the University of Washington, and yeah our institute. So yeah, I want to like thank everybody yeah to help and to support this project, and then yeah. I finally want to thank my lab, my professors, and also the other lab member. I just generated this by Nano Banana. So Nano Banana, I also acknowledge Nano Banana because like yeah, it's a really helpful tool to generate all of this. Yeah, pretty cool figures. Yep. So this is everything. And then yeah, I'm definitely happy to have more questions if you have any questions. Yeah, happy to discuss.
Okay, thank you so much for sharing such an insightful research with us today. So I like that image. That's really
Oh,
nice.
thank you.
Yeah, through a presentation, I was able to learn about the overall concept and research of protein design. So now let's get some questions. So if you have any question, please use the raise hands function on Zoom, and I will call you on your name in order. So let's see. So if you have any question, feel free to raise your hand and ask your questions. 네, 그리고 저희가 수업이 외국인 학생이 보통 있었는데, 인터내셔널이 이번에는
네,
없는 것 같아요. 그래서 원래 이제
네,
퀘스천은 다 한국 학생들은 한국어로 진행을 해도 되고, 네, 편하게
네,
한국어로 답을 하셔도 되고, 영어가 더 편하시면 영어로 답을 하셔도 됩니다. 그래서 첫 번째
네.
학생은 남혜윤 학생 질문해 주세요.
네, 혹시 잘 들리시나요?
네.
그럼 저는 한국어로 질문 드리도록 하겠습니다. 먼저 좋은 강의 너무 감사드리고요. 제가 중간에 놓쳤을 수도 있는데, 알파폴드 3가 주로 이제 한 번 프리딕션 할 때, 약간 싱글 스태틱 스트럭처만 프리딕트 할 수 있는지가 궁금합니다. 왜냐면, 어, 제가 알기로는 단백질이 이제 컨포메이셔널 체인지가 자주 일어나는데 이 알파폴드 3가 이런 것까지 핸들링 할 수 있는지가 궁금합니다.
네, 사실 아웃풋은 싱글 스트럭처지만 이 ML 모델은 그 랜덤니스가 어느 정도는 있거든요. 그래서 뭐 모델링 특성상 막 드롭아웃을 사용하거나 그런 등등의 요소들이 다른 매번 이렇게 프리딕션을 할 때마다 다른 아웃풋을 가져올 수도 있고 그런데 더 중요한 점은 이런 내추럴 프로틴에 대해서는 저희가 항상 MSA라는 Multiple Sequence Alignment, Sequence Information을 같이 넣어주잖아요. 이게 결국에 MSA가 어떻게 이용되냐 하면은 그 되게 유사한 시퀀스들을 주고 이걸 다 얼라인하고 이 포지션과 이 포지션을 봤을 때 얘네가 코일볼빙 했는지 그러니까 하나가 바뀌는데 다른 하나가 바뀌면은 사실 모델이 그럼 얘네는 되게 3D 코디네이트 상에서 가까이 있을 확률이 높다라고 생각하는 거죠. 그게 약간 evolutionary information이잖아요. 모델이 학습하는. 그래서 사실 MSA를 어떻게 넣어주냐에 따라서 스트럭처가 바뀔 때가 있어요. 그래서 그 기존에 많이 이루어졌던 방법들이 이게 어떻게 보면 되게 오토메이션이 되지 않고 되게 프로틴 클래스마다 다르고 사실 어려운 문제인데 이 MSA를 다른 방법으로 뭐 PCA 이런 등등의 방법으로 서브 클러스터링 해서 인풋으로 넣어줬을 때는 컨퍼메이션 A를 만드는데 다른 MSA를 이렇게 서브 샘플링해서 넣어주면 컨퍼메이션 B를 주더라고요. 왜냐면 MSA에서 추출할 수 있는 인포메이션이 다르니까요. 그래서 그런 방법으로 멀티플 컨퍼메이션을 약간 프리딕션 하는 논문들이 여러 개 존재했는데 그 약간 하나의 정립된 방법은 없는 것 같아요. 왜냐면 MSA라는 게 어떤 거는 서브패밀리가 너무 많고 어떤 거는 없고 어떤 거는 그러니까 시퀀스 유사도가 비슷하고 적고 해서 뭐 하나의 딱 그 필터링으로 했을 때 항상 A, B를 추출할 수 있어. 이게 아니고 어떤 프로틴에 대해서는 이렇게 서브샘플링 해야 되고 어떤 프로틴에서 이런 다른 서브셋을 해야 되고 그러니까 다양한 문제들이 존재해가지고 제 생각에는 아직은 딱 정립된 방법은 없지만 그래도 기본적으로 이런 약간 서브샘플링 등을 통해서 모델이 다양한 컨퍼메이션을 추출할 수 있다라고 이해할 수 있습니다. 네.
감사합니다.
네.
네, 다음은 강윤수 학생 질문해 주세요.
잘 들리실까요, 혹시?
네.
네, 발표 너무 잘 들었습니다. 제가 사실 이 분야를 잘 몰라서 좀 여쭤보는 걸 수도 있는데, 이런
네.
그 컨택 레지듀어 예측 같은 걸로 이제 후보들의 결합 부위를 특정할 수 있으면은 그런
네.
정보들을 좀 종합해서 예를 들면 저희가 뭐 목표로 하는 단백질 X의 특정 레지듀어, 예를 들면 세린 300번 이런 거
네.
주변에 좀 선택적으로 결합하는 새로운 스몰 몰레큐를 또 설계하는 게 가능할지, 그리고 또
네.
이런 단계에서 출발해서 실제로 이제 저희가 임상 단계에서 화학적으로 좀 안정적이고 이제 현실적으로 사용할 수 있는 라이간들을 만들기 위해서도 추가적으로 어떤 그 데이터 셋들이 필요한지 이런 것들이 좀 궁금합니다.
네, 그래서 제가 생각하기에 이제는 그게 되게 충분히 가능한 방법이 된 게, 뭐, 세린 뭐 300번에 붙이고 싶어. 어떻게 할까? 할 때, 이제는 이 모든 스트럭처 프리딕션 모델이나 디자인 모델들이 사실 그런 컨디셔닝 메서드에 같이 학습이 돼서 그 인퍼런스, 그러니까 샘플링 할 때, 이 포지션에 붙여줘 하면 그 디퓨전 안에서 사실 뭐 포텐셜이 있어가지고 이 포텐셜이 모든 레지듀를 약간 이쪽 방향을 선호하도록 약간 가져다 주는 그런 약간 모델링의 포스가 있거든요. 그래서 그런 것들을 통해서 사실 컨디셔닝은 하기 되게 쉬워졌어요. 그래서 제가 아까 보여드린 니파 바이러스도 저희가 이미 어디가 인터페이스인지 알잖아요. 그래서 그 사람들이 쓸 때는 모델한테 인풋을 알려준 거야. 이 포지션이 핫스팟인데 여기에 붙여줘라고 말을 하면은 이제 제너레이션 된 애들이 뭐 아닌 경우도 있지만 대다수 그 부위에 약간 결합된 애들로 디자인이 되게 되죠. 그래서 그렇게 네, 접근할 수 있고 그렇게 만들어진 애들을 그럼 어떻게 더 임상적으로 좀 약간 스테이블하고 좋은 애들로 디벨롭 시키냐. 네, 그래서 저는 약간 property optimization이 되게 중요하다고 생각하는데 사실 이렇게 시퀀스만 디자인한다는 거는 얘가 이뮤노제네시티가 있는지 아니면 진짜 인비보에서 스테이블한지 인비보 클리어런스 안 되는지 등등은 저희가 고려할 수 없는 부분이잖아요. 그래서 약간 다른 주제로 사람들이 이런 멀티 디프런 프로퍼티를 옵티마이제이션 할 수 있는 우리 툴을 만들어야 된다. 그러기 위해서는 새로운 데이터가 필요하다. 왜냐면 뭐 어떤 애들이 약간 protease resistant 한지 그런 등등의 약간 정보를 다 알아야지 사실 이런 structure prediction 모델이냐 아니면 PDB 데이터베이스만으로는 그 인포메이션을 알기 어렵잖아요. 그래서 얘가 필드가 나아가고 있는 방향이 우리가 진짜 이렇게 다양한 폴드에 대해서 다양한 프로퍼티를 약간 다 이렇게 실험적으로 추출해서 어떤 그 뭐 그러니까 파운데이션 한 모델을 만들어서 이 시퀀스를 디자인했을 때, 얘 뭐 스테빌리티는 어떤지, 이뮤노제네시티 어떤지, 클리어런스 타임은 어떤지 이런 등등의 것을 더 예측할 수 있으면 더 확실히 약간 드럭 디벨롭먼트에 가까워질 수 있지 않을까라고 생각하고, 네, 그러기 위해선 더 많은 데이터가 필요하다고 생각합니다.
감사합니다.
네,
네, 다음은 박지혜 선생님 질문해 주세요.
네, 안녕하세요. 어, 어, I just wanna trying to use English, so I will use the English. Is
Yep.
that right? Yeah.
Mhm.
Is it okay? Yeah.
Yep, awesome.
Yeah, hello. I'm so impressive and interesting to me. And I'm very appreciated. I have only biological background, not engineering, so it's difficult to understand the contents fully. But I know that, as you said earlier, the AlphaFold3 program is used for prediction some binder protein against target. I have studied some viruses, including influenza virus and vaccine development. Recently, I'm wondering when we designed the vaccine antigens to mimic virus antigen,
Mhm.
in basically when the virus entered the host cells, their surface protein binding to receptor and into the cells, they cleave two parts by the pH dependent manner. So my question is it possible to design consider pH dependent manner using AlphaFold 3 or any machine learning programs, even though any more hallucination and any other errors?
Yeah,
Because
that's a really
yeah,
good question. Mhm.
yeah.
Right, but yeah, I think that's a really good question. But I think yeah, it's also kind of like relevant to this data scarcity problem because I think yeah, the PDB data set like that doesn't really consider the pH, and then yeah, you just like make a right like cell concentration. You just like you need to decrease the temperature. So like sometimes even the structure is like very different with the like natural existing like structure in our body, right? Because like our body have a different temperature, like the different cell concentration, everything like. X-ray crystallization, like sometimes like just force it to like make that like specific conformation in crystal lattice. So I think yeah, to make the model that can be yeah like sensitive enough for that input information, like for example pH and everything, like I think you need to make a right data set like that can understand the difference between the pH. So for example, like measure the like something at pH 7, measure something at pH 5, and then measure the stability, everything, and then like make the model to get a sense about like yeah what actually makes the difference like between the different like pH, because I think AlphaFold model like is not actually solving the protein folding problem. It's actually it actually solve the structure prediction model, which means like it doesn't really understand the whole like this like conformational landscape, everything. So I think yeah, we are like pretty far away. Yeah, how can we understand this like native physics and the interactions and then like fold this state to unfold this state, like what is the pathway between that? So to understand that, like I think yeah, model either need to like understand this like real protein physics, or yeah, you just curate the better data set so the model can extract the better features from the differences between different environment, different pH, and do that. So yeah, the answer solution is yeah, I haven't seen a really good model that is very sensitive enough to figure out the pH difference. So I think my hope is like yeah, if someone can curate a really really large database like that can understand the pH difference, yeah, that'll be really cool.
Yeah,
네,
thank you.
김수빈 학생 질문해 주세요.
네, 들리실까요?
네.
네, 좋은 강연 감사드립니다. 제가 알파폴드에 대해서 잘 몰라서 드리는 질문일 수도 있을 것 같은데, 그 알파폴드를 이용해서 시퀀스 디자인이나 뭐 스트럭처 디자인을 할 때 일반적인 아미노애시드가 아니라 내추럴 아미노애시드를 이용한 디자인도 가능한지 궁금합니다.
네, 그것도 모든 건 사실 데이터 프로블럼인데 PDB를 보면은 unnatural amino acid 그 사실 unnatural amino acid가 되게 니시한 파트잖아요. 그러니까 뭐 전 세계 과학자가 막 1000명이 있으면은 한 뭐 10명 정도만 그거를 공부하니까 결국 PDB를 보면은 한 1000개 중에 한 개에서 10개 정도만 unnatural protein이어서 일단 데이터가 되게 작아요. 그래서 저도 최근에 관심 있는 게 D-amino acid prediction인데 D-amino acid가 mirror image amino acid잖아요. 그래서 되게 어플리케이션도 많고 사실 드럭으로 디벨롭 할 때도 디아미노 아시드가 약간 protease resistant 하기 때문에 되게 좋은 타겟인데 아 진짜 디아미노 아시드 structure prediction 하는 모델을 만들고 싶다라고 생각을 했지만 PDB를 보면은 이 250K의 스트럭처 중에서 1500개만 디아미노 아시드인 거예요. 그것도 되게 스펄스하게 디아미노 아시드가 있는 그러니까 거의 약간 1%도 안 되는 그 데이터셋으로 모델이 충분히 그걸 언더스탠딩 할 수 있게 만들 수 있을까를 했을 때 아직은 엄청 이런 L 프로틴처럼 좋은 accuracy에 도달하지 못한 것 같고 그래도 모든 모델들이 디아미노시드에 트레이닝을 하기는 했어요. 그래서 인풋으로 사실 줄 수는 있고 뭐 이렇게 X 토큰에 디아미노시드 뭐 어떤 사이드체인이 다 있는지 그 스페시파이 할 수 있고 그다음에 뭐 포스포릴레이션 뭐 이런 것들도 뭐 세린 포스포릴레이션, 드레오닌 포스포릴레이션 이런 것도 스페시파이를 해서 모델이 프리딕션을 할 수 있어요. 단 주의해야 될 점은 이게 진짜인지 아닌지는 의심을 해봐야 된다. 이게 뭐 뭔가 프리딕션을 했다 해서 이게 완전 그라운드 트루스구나라고 생각을 하면 되고 당연히 모델이 못 이해한 부분도 있기 때문에 이 스트럭처가 맞는지 아닌지를 약간 충분히 검토를 하고 사용하는 것이 좋다고 생각하고 앞으로 네 그런 쪽의 방향으로 네 더 나아가야 된다고 생각합니다.
감사합니다.
네.
네, 마지막으로 천호서 학생 질문해 주세요.
네, 잘 들리시나요?
네.
네, 앞선 좀 부분에서 되게 알파볼드에 대해서 질문을 많이 하셔서 저는 그냥 프로틴 홀딩 자체에서만 좀 포커스를 해서 질문 하나 드리고 싶은데 두 개인가? 아니, 하나만 질문 드릴게요. 그러니까 프로틴 폴딩이라고 하면은 조금 그냥 깁스프리 에너지가 가장 낮은 미니멈을 좀 이제 가장 옵티멀한 홀딩이라고 보는 것 같은데 지금 보니까 이게 뭔가 조금 얼핏 제가 잘못 들은 걸 수도 있는데 듣기로는 좀 로컬 미니멈 그런 좀 스팟이 존재를 하는 것 같아요. 그럼 이제 로컬 미니멈이 이제 존재를 하는 이유는 잘은 모르겠지만 에너지 배리어가 높다던가 그거를 잘 못 높다던가 아니면 케네디 그런 게 좀 있을 것 같은데 이제 이게 이 로컬 미니멈이 글로벌 미니멈이 될 수도 있는 건가? 조금 생각이 들었는데 그게 맞는 생각인지 잘 모르겠습니다. 맞는
음.
질문인지도 잘 모르겠는데
그리고 로컬 미니멈이 글로벌 미니멈이 되려면 그 에너지 자체가 앱솔루트 에너지가 가장 낮아야겠죠? 네, 그래서 그냥 여러 개의 약간 로컬 미니머가 있으면 그거에서도 사실 약간 비교했을 때 뭐가 더 높고 낮음을 알 수 있잖아요. 사실 저희가 이게 그 델타 G라는 걸 생각해 봤을 때 마이너스 RTL은 unfolded versus folded니까 이게 모든 게 다 equilibrium state에 있는 거잖아요. 그래서 예를 들면 1000개의 프로틴이 있을 때 저희는 뭔가 1000개 다 folded 돼 있는 게 아니고 사실 얘의 그 에너지 랜드스케이프를 알면은 어떤 게 로컬 미니머고 뭐 어떤 게 막 글로벌 미니머고 등등을 고려했을 때 아마 60개 정도는 이렇게 하나의 타임 프레임을 찍었을 때 60개 정도는 여기에 폴딩이 돼 있고 뭐 20개 정도는 다른 로컬 미니어 폴딩이 돼 있고 그러니까 사실 이게 모든 게 다 이퀄리브리엄 스테이트여서 네 당연히 프로틴은 다 이퀄리브리엄 스테이트에 존재하고 여러 개의 로컬 미니머가 있을 수 있고 그거는 다 그런 이 relative difference에 의해서 결정이 된다라고 말을 할 수 있을 것 같아요. 그래서 대부분의 프로틴은 사실 점점 커지면 커질수록 이 투 스테이트만 존재하는 경우가 되게 드물어요. 투 스테이트라는 거는 완전 unfolded. 그래서 제가 말했던 아까 스트링처럼 존재하는 거 versus 약간 fully folded 그렇게 존재하는 것보다 그러니까 멀티 스텝이 있거든요. 그래서 그 멀티 스테이트가 있어서 confirmation A, B, C 그러니까 막 이게 완전히 다른 게 아니더라도 약간 partially unfolded 된 거, fully folded 된 거 약간 이런 식으로 다 존재를 해서 그게 다 퍼널 형식으로 구성돼 있는데 네 점점 커지고 이게 프로테인이 커지면 커질수록 그걸 사실 시뮬레이션 하기도 너무 힘들고 시뮬레이션 한다라고 하면 뭐 MD 시뮬레이션이나 이런 걸 하는데 그런 걸로 약간 스테이트를 정의하기도 어렵고 이 스트럭처 프리딕션 모델 아까 제가 말했듯이 뭐 스토캐스틱 하긴 하지만 그래도 그런 어떻게 랜덤니스를 주느냐에 따라서 스트럭처 프리딕션이 너무 다르기도 하고 그 스트럭처 프리딕션은 프리퀀시 다른 프리딕션 할 때마다 다르게 나오는 것들 프리퀀시가 사실 이퀄리브리엄을 대변하진 않기 때문에 그거를 약간 프록시로 사용하기는 어렵다라고 볼 수 있고 혹시 답변이 되었을까요?
네, 감사합니다.
네, 질문이 다 없는 것 같아요. 제가 마지막 하나만 정말 기술적으로 저도 잘 몰라갖고 질문 하나 드리고
네,
싶은데요. 제가 원하는 타겟 프로틴이 만약 되게
네.
좀 컴플렉스한 뭐 멀티 다이머릭, 헤테로 다이머릭한 것도 그것에 대한 바인더도 쉽게 예상이 가능한가요?
네, 그게 사실 타겟마다 정말 다르긴 해요. 왜냐면은 아무리 프리딕션이 잘 돼도 실제로 해보면 안 되는 경우들이 꽤 있거든요. 그래서 뭐 이게 뭐 막, 무조건 잘 되고 무조건 안 되고 이렇게는 할 수가 없지만 그래도 앞으로 점점 나아가고 있는 방향이 프리딕션 퀄리티가 좋아지면서 그에 맞춰서 디자인 퀄리티도 좋아지고 충분히 멀티머릭 프로틴을 줬을 때 프리딕션 하면서 동시에 그 바인딩도 예측을 할 수 있는 그 애큐러시가 점점 높아지니까 그냥 제가 말씀드릴 수 있는 거는 점점 모델들이 좋아지고 있다. 네, 점점 아마도 이 모델들로 만들면은 더 그런 인실리코 석세스 레이트뿐만 아니라 실제로 실험에서도 더 웰랩 석세스 레이트도 점점 높아지는 방향으로 가고 있다고는 말씀할 수 있지만 그 프로틴 어떤 프로틴이냐에 따라서 다른 것 같은데 기본적으로
음.
되게 어려운 프로틴은 또 다이나믹한 프로틴이에요. 그래서 프로틴이 인터페이스가 되게 바뀌는데 이 컨퍼메이션 A를 약간 이렇게 락인 해가지고 이걸 스테빌라이즈 하고 B로 안 바뀌게 하고 뭐 그런 등등의 되게 어려운 인터페이스를 다루는 건 아직까지 되게 조금 힘든 문제라 여겨지지만 되게 프로틴 자체가 스태틱하고 딱 그 어디가 하이드로필릭 패치인지 하이드로포빅 패치인지 그거에 되게 명확한 프로틴에 대해서는 비교적 쉬운 문제인 것 같아요.
아, 네. 박지혜 학생이 질문 마지막 짧게 해주실래요? 시간이 많이 지나서.
네, 죄송합니다. 아까 니파 바이러스에 대한 글라이코프로테인 프리딕션 해주신 거 보여주셨었는데, 저가
네.
바이러스 하다 보니까 좀 더 궁금해지는 게, 그러면 니파 바이러스 프리딕션 하실 때는 리셉터의 그 바인딩 사이트를 기반으로 프리딕션 하신 건가요?
네, 맞아요. 그래서 리세터 그쪽 인터페이스, 그러니까 PDB에 사실 그 프로틴하고 에프린 자체의 컴플렉스가 있어서 그 두 개의 인터페이스를 이 레지듀 사이의 간격을 통해서 어떤 게 바이딩한 레지듀인지 다 인포메이션 추출하고 그거를 이제 인풋으로 넣어준 거예요.
아, 그렇구나. 네, 감사합니다.
네,
네, so we will wrap up today's class. Once again, a huge thank you to Yerin Cho for sharing her research and expertise with us. And thank you to all students and questions. And we will see you again next week. 감사합니다, 박사님.
네,
졸업 이제 곧
감사합니다.
축하드립니다. 네, 다음에
네,
또
감사합니다.
뵙겠습니다.
네,
네.
좋은 하루 보내세요.